blood-brain barrier. It is a highly selective semipermeable barrier protecting the brain and spinal cord from harmful substances. It allows oxygen, glucose, and essential nutrients to pass through the membrane but does not allow the passage of toxins, pathogens, many drugs, etc. This helps maintain a stable environment for neurons. So the functions of blood-brain barrier include protection from toxins, CNS hemostasis, prevents infection, regulates drug entry, etc. The structure of blood-brain barrier includes capillary endothelial cells that are connected to each other by tight junctions, a basement membrane on which the endothelial cells are present, and the food processes of astrocytes. These tight junctions are important because they prevent passage of substances between the endothelial cells, thereby maintaining the selective permeability of the blood-brain barrier. The basement membrane provides structural support. The food processes are small swellings at the ends of astrocyte. Astrocytes are neuroglial cells. They adhere to the outer surface of the capillary wall. Certain areas of the brain lacks blood-brain barrier. These are called circumventricular organs. The examples include pineal gland, posterior lobe of pituitary gland also called as neurohypophysis, area postrema, subfornical organ, organum vasculosum of the lamina terminalis, median eminence located at the base of hypothalamus, etc. There are actually reasons why blood-brain barrier is absent in these areas. The pineal gland secretes melatonin directly into the blood to regulate circadian rhythm and sleep-wake cycle. The neurohypophysis releases hormones like ADH and oxytocin directly into the bloodstream. The area postrema detects toxins or harmful substances in blood and triggers vomiting reflex. So these areas lack blood-brain barrier because they must exchange substances rapidly with blood for proper physiological function. If a tight blood-brain barrier is present, sensing of blood changes or secretion of hormones would become insufficient. In patients with Parkinson's disease, there is a deficiency of the neurotransmitter dopamine in the substantia nigra. But dopamine cannot be given because it cannot cross the blood-brain barrier. Instead, levodopa, that is L-dopa, which is a precursor of dopamine, is given. It crosses the blood-brain barrier and converts into dopamine by the neurons within CNS. Normally, many antibiotics like penicillin cannot easily cross the blood-brain barrier. But in meningitis, the blood-brain barrier is broken down and this allows high concentration of penicillin to enter CNS. This is clinically beneficial in treating the infection. Damage to blood-brain barrier causes leakage of fluid into the brain leading to vasogenic edema. This damage can be due to trauma, tumors, infection, or hypertension. Kernicterus, also called as bilirubin encephalopathy, is a severe form of jaundice that is seen only in the newborns. It occurs when unconjugated bilirubin crosses the blood-brain barrier and deposits in the brain tissue. This is because the blood-brain barrier is immature and not fully developed in the newborn. Bilirubin becomes toxic to the neurons, resulting in neuronal damage and encephalopathy. And that wraps up a high-yield overview of blood-brain barrier.